Welcome, everybody. This is our Friday afternoon seminar series, and today we welcome Kelly Zamudio from Cornell. It's my great pleasure to introduce her. When you peruse the literature like I do, you see Kelly's name all over the place because she has worked on so many different levels in biology, and that's characteristic of her entire career. And although I didn't know her when I was at the University of Washington because she followed me, she worked with the most important intellectual mentor I had, Ray Huey. And so I looked at what she was doing, and I knew that both Liam and Doug would be very interested in having her come. And so I emailed them and said, oh yeah, we'd love to interact with Kelly. So here we are. A little bit of Kelly's background. She did her undergraduate work at Berkeley and got a NSF, a prestigious NSF fellowship, a predoctoral fellowship to go to the University of Washington, where she did her work on lizards. And her thesis is characteristic of her whole career in that she used many different approaches. She did field work and experimental work, she did lab work, she did a phylogenetic analysis, and she did conservation work, all in the same thesis. It's really remarkable. And so it's not surprising that she went on to continue collaborating and doing incredible work on a variety of herps. She has over 150 publications. She's been elected to the American Academy of Arts and Sciences. She holds the Stephen H. Weiss Presidential Fellowship Award at Cornell. And so we're just and she's the Gordon Smith Professor of Ecology and Evolutionary Biology and the curator of herpetology. So she has incredible responsibilities. On top of all that, which is dear to the hearts at UMass Boston, is that she's done a lot with education, particularly active learning, which we know is very important for students. And so we'll see if she uses any of those techniques during her lecture, whether she reverts to her standard lecture mode that we were all taught with.
Oh no, a challenge.
Challenge. Anyway, we welcome you today and to learn both about the basic biology. She's always doing a lot of basic biology and conservation at the same time. So it's exciting to welcome you to UMass Boston, Kelly.
Thank you so much. What a nice introduction. I really appreciate the invite. I'm excited to share with you guys some stuff that we've been doing in our lab. And I've got a little bit for everybody, a little bit for Doug and a little bit for Liam. And well, we'll see what happens. I probably revert. Webinar format is not so great for active learning interactions after all, but we'll see what happens. Okay, so let me tell you a little bit about some of the work that's been going on in our lab. I wanted to focus on two very different sides of the lab work, just because I sort of feel like I run these two tracks in parallel. And one is asking questions about how frogs diversify, and specifically in terms of how they reproduce. And then the second part of the talk will be about some of the work we've been doing on the chytrid fungus that is causing the frog pandemic and declines worldwide, which you guys are all familiar with. Okay, so let's get started. First, I'm going to talk about sexual selection and mating systems in frogs. Frog reproductive modes have been something that have been studied, oh my goodness, for probably over 100 years. People have been interested in that. And the reason is because they're so diverse, right? So the frogs, I always like to say in my herpetology class that they sort of reproduce in all ways possible, and they give birth to babies, and they drop their eggs out of pretty much every orifice in their body, maybe with the exception of the ear, right? So the reason I say that is because if you think about sort of frogs and where they put their eggs, and well, if you think about frogs and their life histories, right, they often put eggs in a particular environment, often aquatic, but not always. The tadpoles have to develop in a particular place, and that microhabitat is important. And then after metamorphosis, when the tadpole becomes a juvenile frog, if that is part of the species life history, there can be parental care of the juveniles or of the eggs or or even of the tadpoles, right? So if you think about all the different places where a frog can make a decision to put an egg, to put a tadpole, or to take care of the different life stages, you can see how sort of reproductive modes cumulatively become so diverse because there's just so many different stages where evolutionary decisions are made. So when we think about frogs putting eggs, Around here in the temperate zone, we usually think about what we know to be the ancestral state, which is frogs putting their eggs in water in a pond or in a stream, like this European frog here. But there's a lot of diversity. This leptodactylid from South America puts eggs right at the edge of the water, right where the grass meets the water at the edge of the pond, and creates a foam nest to protect the eggs from desiccation. Other species remove their eggs altogether from water, either by putting them on the surface of leaves overhanging water, or by finding little containers of water like the axillas of bromeliads or bamboo stalks, where the eggs can be hidden from the larger water body. And then some frogs like this one here in Brazil actually combines that. So these eggs here are in a foam nest inside of an axilla of a bromeliad. So you get the idea. Basically, if you have all this variation at every life stage and then you add a bunch of life stages, you can end up with a lot of diversity. Here's some of the examples that I like the most because they sort of show a shift towards terrestriality. The top figure here is a hylid that creates this little mud basin, and then the male stays and guards basically his own little private pond. And the one on the bottom creates a little volcano from which he calls and attracts females. The females enter the volcano with the male, and amplexus and egg laying all happens inside of this sort of private volcano-like chamber. All right, so that's sort of an introduction to reproductive modes. Now, why am I particularly interested in this trait? Well, I'm interested in this trait because it just fascinates me that there can be different selective pressures or environments at each of these different life stages. And I think that has a lot to do with explaining why there is so much diversity. Okay, so a few years ago, this paper came out by Gomez Mestre et al. In 2013. And it was a really nice data set. Basically, they took all amphibians that they could find on the tree of life, and they categorized what they called frog reproductive modes on the tree, and asked questions about how labile transitions between these different reproductive modes were. Now, just to back up a little bit, the dogma, I think, in the past about sort of reproductive modes in frogs is that, you know, we had this ancestral condition, which is primarily aquatic eggs, primarily aquatic tadpoles, metamorphosis, and adults go into a terrestrial environment. And the idea in the past is that all these derived, sort of more specialized, more terrestrial or hidden eggs or building a nest, all these more derived characteristics or traits were sort of embellishments on a theme, right? And the main selective advantage that was proposed for these changes in reproductive mode had to do with getting your tadpoles out of the water. It's what they call the predator escape hypothesis. And the idea was that frogs have a tendency, or over the evolution of the frog tree of life, the tendency to move tadpoles or eggs out of water had to do with getting your tadpoles away from the predators that were in the pond. So Gomez Mestre were actually testing that. And in fact, they found that there were numerous cases in the frog tree of life with specializations that involved terrestriality at some life stage, that most of those derived states, more terrestrial states, tended to be in the tropics. And they found, using this analysis here, that there was a lot of sort of movement back and forth. Okay, it wasn't just a chain of increasing terrestriality. There was multiple evolutions on the tree of different forms of increased terrestriality and reproductive modes. So I looked at this and I thought, well, this is okay. This is good. But there was something that was really bugging me about this, and I couldn't figure it out for a while. And then I finally figured out that what was bugging me was that the way that they coded their reproductive modes already assumed that the tadpole and the egg were somehow correlated in terms of where they could be based on the predator escape hypothesis. So if you look at the 4 states in this diagram here from their paper, you can see that they've already tied the protection of egg with the protection of larvae. So you can either have unprotected egg and unprotected tadpoles. You can protect your eggs, but not your tadpoles. You can protect both your eggs and your tadpoles, or you can go fully terrestrial and have direct development. So you see what I'm saying? There's many reasons that a frog might want to put, potentially, eggs on land. A species might want to put eggs on land but have an it vice versa. And by coding the traits this way, it it's almost like you're automatically presupposing that the predatory escape hypothesis is what's driving all this variation. So I and my colleagues from Brazil decided, who have been studying reproductive modes in the tropics for a long time, decided that we would try to reanalyze this by trying to decouple and look in a little more detail to see what was going on at the different life stages. And the reason that we did this is that we thought that there might be not just a role for natural selection in the escape from predation, but that there might be some role of sexual selection in terms of reproductive modes, right? Now, the reason we thought that is because there are a number of mating systems that we observe, not only in the tropics, in the temperate zone as well, where there's stiff competition between males for access to female amplexes, and a lot of competition that ends up meaning that males lose reproductive success due to polyandry, meaning they lose fertilization of some of the eggs during this competition. So here's an example. This is our widespread North American plains toad. And you can see right here, there's a calling male right there, vocalizing, attracting females. And right here, you have a satellite male that is quietly waiting until a female comes in so that he can try to amplex her as well. There are some more extreme examples. This is a group breeding frog and from the Pantanal of Brazil. You have an amplecting pair right here. You can see it. They've created this little basin and they're amplecting, and the female is depositing her eggs and the male is fertilizing those eggs and creating this faux nest. And then you have all these not lonely, I guess they're not lonely here, but you have all these independent males with no females that are just putting their sperm into this nest with hopes that their sperm will encounter some of that female's eggs. So you can see this is clearly a high, potentially mating system that would lead to potentially high polyandry. Then we have some other examples like scramble competition. This is a hylid from Brazil, and this is what we call a mating ball, and there's about 7 males, and somewhere in the middle there is a female, and they're all attempting to fertilize her eggs. So there's a lot going on here, right? And so we thought maybe getting out of the water has maybe less to do with just escaping predators, and perhaps it has something to do with mitigating the competition that happens, especially during reproductive season. So to test this, we need 2 things, right, to test this hypothesis. The first is we would need to show that evolutionarily there's a correlation between terrestrial reproduction and somehow avoiding this competition. And one way that frogs avoid this competition is by hiding their amplexus, meaning by getting away from other males and from access, or reducing the access of other males to the females that they're courting. The second thing that we would need to show is that, in fact, hiding yourself during amplexus, you and your pair, reduces the total amount of polyandry, right? So that would be the mechanism that would favor, the selective mechanism that would favor this move to terrestriality if sexual selection is the driving force. So what do I mean by hidden amplexus? So let's go back to these 2 examples here. Here on the left, this hylid has moved terrestrially, but has not completely hidden amplexus, okay? Other males, and we see this a lot, other males of this particular species will come along and try to compete with this male for the females that come to his pond, to his nest. In this case, this is a hidden amplexus, right? There's a limitation of the access of other males to the amplecting pair. Okay. Great. So we decided to test this in a family of ground-dwelling frogs that are called the leptodactylids. There's about 200 species. They're primarily tropical, and they are great because they have a huge variation in terms of what they do with their eggs and where their tadpoles develop. So you have everything from Phylogenetic, the one I showed you before, that breeds basically right at the edge of the pond, to species who create different kinds of burrows, like semi-interred burrows or burrows that are very deep, all the way down to this species here, Leptodactylus phalanx, that basically creates a really deep chamber, deposits eggs. The tadpoles develop entirely terrestrial inside the chamber until metamorphosis when they crawl out as little froglets. So we collected data. First, we reconstructed a tree for the species of leptodactylids, for which we had reproductive data. We collected data on the aspects of reproductive mode we were interested in, did some ancestral state reconstructions, and also used tests to try to ask questions about which characters were actually correlated on the tree, were evolving in some sort of correlated fashion. All right, so I'm just going to cut to the chase. We found some interesting patterns. The first one is that, as we suspected, the tadpole stage, where the tadpole develops and where the egg is placed by males and females reproducing in leptodactylus species, is completely disassociated evolutionarily. Okay, so this idea that getting your tadpoles and your eggs out of water to avoid predation, it just doesn't hold based on this complete disassociation between those two. Okay, so the fact that they don't evolve in any correlated fashion means that different selective pressures could be acting in those two different life stages. We also looked at where species put their eggs mattered in terms of a correlation with whether they had parental care or not, and we found no association there evolutionarily. The only two characteristics that were correlated evolutionarily on the tree was where species put their eggs and whether the amplexus is exposed or hidden. And those two traits, at least for leptodactylids, evolved dependently, meaning they're associated evolutionarily on the tree. This is the output from the correlation analysis, the base traits analysis that we did. And basically these solid arrows indicate, okay, wait, let me back up a little bit. The four boxes include all possible combination of that pair of traits, whether the egg is aquatic or terrestrial, and whether the amplexus in that species is exposed or hidden. And the way to interpret this is that these states, these correlated states, can vary in all possible combinations, and the solid lines, the solid arrows, indicate transitions that are significant. And so here, for example, in species that have exposed amplexus, if changes are to happen, they tend to be in the direction of terrestrial, of going towards aquatic eggs, okay? Over here, if amplexus is hidden, the correlated changes on the tree are going to happen in the direction towards terrestrial eggs, okay? So basically what this means is that evolution, at least in terms of signatures on the phylogeny, hidden amplexus is strongly correlated with terrestrial eggs, and exposed amplexus tends to be correlated with aquatic eggs, or at least transitions in those pairs of traits tend to be correlated on the tree. So that's cool. That means that frogs, that eggs and tadpoles probably aren't actually responding in terms of to the same selective pressures in terms of degree of terrestriality. So we could show that there's that evolutionary association. Can we actually show that moving onto land and hiding your amplexus really does reduce polyandry? Well, I wish we could. If I had a wish, it would be to actually be able to measure the degree of polyandry in all 200 species of leptodactylids. I don't know if that'll take a few careers to do that, but what we can do is try to get at this question sort of in a different way. And that is to use testis size as a proxy for polyandry. So the size of a frog's testis, right here, this is a testis that's been dissected from this frog. The size of the frog's testis is correlated with the rate of production of sperm. And frogs that engage in mating systems that have high potential for polyandry usually involve high potential for sperm competition. And so those frogs tend to have larger testes, especially if you adjust for the size of the male's body size, right? So we can use this as a proxy and ask whether species with different kinds of amplexus, hidden or exposed, actually show what you would expect in terms of response in testis size. Okay, so we used measured testis size from a number of different species. This one right here is the frog that I told you about before that mates communally or in a group. And here is what we found. So let me walk you through this. The y-axis here is the percent testis weight adjusted for male body size. These are all the species with exposed amplexus, and it's hard to see here, but there's about 17 points here, and these are all the species with hidden amplexus. Okay, so relative testis size, in fact, was different between the two groups, but in the direction that we predicted. Frogs with hidden amplexus have tiny testes. Okay, and taxa, as you might predict as well, with exposed amplexus have much higher variance in testis size, and some of the largest testis sized species are the ones are in this category with exposed amplexus. Now, these data are preliminary. We still need to get a lot more data on testis size in the other species, and also we need to correct this analysis phylogenetically, which we haven't been able to do yet because I only have 17 of these hidden species so far. But one thing that's interesting to me is, one, these outliers. Clearly there's something incredible going on there, right? And in some of the cases, we know why these numbers are so large. But another interesting thing to me is what's going on down here. If these are frogs with completely exposed amplexus and they aren't faced with a challenge in terms of sperm competition, what else is going on either behaviorally or in some other aspect that is causing this pattern? So I think these guys might actually be worth investigating. Okay. All right. So all of this to say that I don't think this all has to do with predation, the evolution of this diversity in reproductive modes. And my bet is that it probably doesn't all have to do with predation. Not that predation doesn't matter. I'm not saying that at all, but I do think that there's more in terms of mechanisms to explain this diversity. And what this study did show us is that we need a lot more observations on species-specific characteristics and traits to really get at some of these selective mechanisms. And so we have plans to sort of go through a number of species, focusing especially on certain clades that have a lot of reproductive mode diversity, and complementing this work with field studies to sort of ground truth some of this. Okay, great. All right, now I'm going to switch and talk a little bit about our disease work. The two are actually related in some interesting ways because it turns out that reproductive modes actually influence the transmission of chytrid fungi and frogs in some interesting ways. And so it's kind of fun to sort of see these two aspects of our lab coalesce in some unpredicted ways. But okay, so I study chytrid fungi of amphibian. There are two species out there that we know of right now that are obligate parasites on amphibian skin. One of them is BD, and the other one is called Batrachochytrium salamandrivorans, B-sal. You guys probably know about these parasites because there's a lot of work going on in your department on them, but basically they are incredible pathogens. Okay, it's like you couldn't make a better pathogen for an amphibian, right? They love keratin, which is present in frog skin. They love being in moist and aquatic habitats, which frogs like too. They move in the water, so transmission is fairly easy in aquatic habitats, which most frogs use at some point in their lives. BD is here and distributed pretty much globally. And B-sal is actually in Western Europe. We think it came from Asia, and basically, as far as we know, has not arrived in the US yet. We do have it in the lab in the US, but it hasn't arrived and it is not yet pathogenic to our local salamanders. All right. So everybody always asks us these two questions. What's the impact on amphibian diversity, and where did this thing come from? Where did BD come from? So I'm going to talk about BD first, since we know more about it. A couple of years ago, a bunch of us got together and put together a database of all known cases of frogs where there was some evidence that BD had contributed to declines of some severity. Okay, so not all declines are severe. You can see the categories of decline here. Some species are affected with less than 20% of their population. Some are in between. And then these hotter colors here represent species that are severely affected. Okay, so more than 90% of the inferred declines on the level of all populations, or species that are presumed to be extinct or that have become extinct. And you can see that the impact is not even globally. Okay, there seems to be huge impact in Central America, a large impact in South America, and I've separated Brazil from the rest of South America here just to show that South America's impact is relatively high outside of Brazil, and that's because of the declines in the Andean species. We have some declines here in the US and some in Australia, and then we have fewer in Europe, and then basically a lack of a lot of information from Africa. Okay, so this is the impact that we see. So we estimated approximately 500 species impacted by BD, with a good 20% of those, almost 20%, in these high extinction categories, either extinct or presumed extinct. So we also see some patterns that are expected in terms of impact globally that you would predict as a conservation biologist. Okay, so most of the extinctions happened, most of the severe declines and extinctions happened before BD was even discovered. Okay, so BD was characterized in 1999 by Joyce Longcore and described as a pathogen of amphibians. We also find that most of the species that are most severely impacted are actually species with smaller range sizes, just as you would predict. So here, these concentric circles represent smaller and smaller range sizes as you get to the middle, and you can see that the hotter colors, the more impacted species tend to have small ranges. The species that are most impacted by BD also tend to have smaller elevational ranges, okay, when they occur along any elevational gradient. And so more severely impacted species with smaller gradients. So basically all of this is pointing to species that are microendemic that maybe have sort of finer habitat requirements or specific habitat requirements are the ones that are going to get hit the hardest. The other question, which we answered, is where did it come from? So we put together a panel of 234 BD isolates collected from throughout the world. This was a large collaboration with a number of people. And we created a phylogeny and tried to understand how BD has differentiated globally. And so here's the tree. And the first thing I want to point out to you on this tree is that there are these three clades here at the base of the tree, blue, pink, and red, which are what we call the enzootic clades. Okay, so there are clades that are restricted to one particular place geographically, including Brazil. The pink one here is what we call BD Brazil, which is one of the strains that I have been working on in Brazil, and I'll talk a little bit about that in just a bit. But these clades that diverged early in the history of BD, in all three cases, we know that these, I think all three cases, no, in two of the three cases that have been tested, we know that those particular clades have less virulence. They're less pathogenic than this large green clade that pops out here in the most derived place in this phylogeny. This large, shallowly diverged clade of BD right here is what we call the global panzootic lineage. It's the lineage that has moved globally, is the most infectious, and is typically associated with the large declines that are reported worldwide. Okay, from this phylogeny, we can infer some interesting things about the genetic diversity of these four different clades. And what you can see is that most of the polymorphism in BD is concentrated in Asia, and that the lineages that are present on other continents are actually subsets of that genetic diversity. So we think that BD as Bisal, as with Bisal, sort of emerged out of Asia as a pathogen, and that this global panzootic lineage is at some point moved to become, or evolved to become more virulent and moved across the whole world. My lab has been working on BD population, BD host pathogen dynamics in a number of different places, primarily in the New World. And the reason that this is fun is because in different places of the world, the hosts and the pathogen have different dynamics. So we know, for example, that in North America, the pathogen is already enzootic, meaning it has been here a while and is basically reached an equilibrium with its hosts, at least in most cases. In Central America, over the course of the last 2 decades, we actually witnessed a number of these catastrophic declines. So we call that an epizootic phenomenon. And then in South America, we believe there are past demographic collapses as well, but there's also the presence of these 2 lineages, the BD Brazil and the BD global panzootic lineage, which makes for some really fun comparisons. Okay, a lot of the studies on BD that go on in my lab have to do with differential gene expression studies, or sort of looking at the functional response of either the pathogen or the host to the infection process and what that means in terms of disease outcome. And we tend to focus on the pathogen itself, its different life history stages, the hosts themselves, because we know that different hosts have different susceptibilities to the pathogen, or we focus on the environment and how that might modulate this interaction between host and pathogen. So I'm going to talk about the 2 studies here that are checked in green. The first is a comparison of salamanders in North America that are either infected with one pathogen, just BD, or are infected with B-Sal. And the second study is not published yet, but I'm going to tell you about a test that we did on a Brazilian frog, so a native host that is infected by the two different lineages of BD that are present in Brazil, BD Brazil and the BD introduced BD GPL. Okay, so let's start up here. This study I think is very timely because, as I mentioned before, B-Sal is not in the wild here in the US, as far as we know yet. But many of our local salamanders actually do carry BD. Okay, so BD tends to have a relatively mild effect on salamanders in terms of pathogenesis. And many of our salamanders, like this eastern newt right here, are actually carriers of BD. They carry it at relatively low loads, but they have it. And so the question that's important here is what happens if our salamanders become co-infected? Now, things could go a couple different ways, right? You could imagine a situation where co-infection by 2 species of the same genus of pathogen could be antagonistic. They're both skin pathogens. Maybe they compete for space. Maybe one suppresses the other. And that would be a good thing, right? It might even decrease disease severity. On the other hand, 2 pathogens infecting the same host might have the opposite effect. They maybe have a synergistic effect or an additive effect. Independent of whatever it is, the bottom line is that that's going to be a poor outcome, right? The disease severity, you could imagine, would increase. So this is important to know, given that most of the infections with B-Sal that we think are going to happen once this chytrid arrives are actually going to happen in the form of a co-infection. So my student, Ana Longo, who was a postdoc with Karen Lips at the time, did this experiment, and we collaborated in terms of the functional genomic responses, but she brought in a bunch of newts from the wild, infected them only with BD, only with B-Sal, or with both. Okay, and then she compared those, and we could compare those to the controls, which were uninfected individuals. She did a lab exposure, and then once the infection trial was over, we collected tissues for the differential gene expression. Okay, so here's what we found. Let me walk you through this. The first box here is what happens when salamanders are infected only with BD. The second is only with B-Sal. And the third is what happens when they are co-infected. These are called volcano plots. And what you're looking at here is basically every single dot here is a gene that we found expressed when we did our RNA sequencing. And the ones that are color-coded, red for upregulated or blue for downregulated, show you the genes that are significantly differentially expressed from the control group. Okay, so they're out of bounds in terms of their expression. And you can see that when salamanders are infected with BD, few genes get upregulated. Not a big deal, right? Salamanders live or seem to tolerate BD well. If you infect them with B-Sal, you start seeing the upregulation of a huge number of genes. And if you infect them with both, as in this case, then you see a huge amount of not only upregulation of different genes, but a large number of genes that are downregulated, right? That have their expression suppressed. This was our first clue that something would be going on here that might not be so great in terms of outcome. And what we can do now is actually query these genes right here and figure out what are those genes actually doing. And so I've put most of the genes that were up or down regulated here, and I can actually, you can just see from this heat map that it's basically like a mirror image of each other. So everybody over here in yellow is in the control group, uninfected. Everybody here in blue is co-infected, okay? So both pathogens together. And there's this huge block of genes here that are underexpressed in the control and overexpressed in the co-infected salamanders. And then there's this other huge block of genes that are overexpressed in the controls, but underexpressed in the co-infected salamanders. So what are these? These that down here that are underexpressed, that have suppressed gene expression in the co-infected salamanders, include among them a number of genes that are part of the complement innate immune response. Complement is part of the first response upon infection. And if we look at where those are, you can see the complement activation, protein activation, bunch of cascades that have to do with immune response, innate immune response. But also importantly, there's also genes in this block here that are underexpressed that have to do with the humoral immune response. This is the immune response that's adaptive, that's a response that eventually results in immunity in a well-functioning immune system. Okay, so that's what's happening there. Up here, what's happening up here? These are genes, remember, that are underexpressed in the controls and then get upregulated in the co-infected salamanders. What we see here is sort of what you would expect, right? We see a number of different gene functional categories that have to do with antigen presentation. Sort of the first steps upon infection for the adaptive immune response is for the antigen to be to enter this cascade. So that's exactly what you would expect upon infection. But we also see a number of gene functional response groups that have to do with signatures of inflammation and innate immune responses. Okay, so this is sort of what you would expect upon first for a vertebrate immune system in terms of first response. So what's happening is that the genes that are supposed to be signaling for something to start happening are actually working as you would have predicted, right? A salamander gets co-infected and starts sending the signals of infection. However, things don't follow through. And everything is suppressed, not only in terms of the innate system, but also the adaptive immune response. So co-infected salamanders are basically severely immunosuppressed and not just in one system, in one immune system, in both the innate and the adaptive immune system. You can look at individual genes in these different systems and the pattern is always sort of the same. Okay, so this is the control right here in the first column. These are salamanders with BD only. This is salamander with B-sal only, and this is co-infected salamanders. And you can see the same pattern in terms of gene expression in each case. What's happening is that the co-infected salamanders are responding to a co-infection as if they were only infected with BD, meaning their gene expression seems to be at that same level. Now, what's going on here, right? There's this downregulation of these really important immune genes. We see this both in the skin. I showed you the information for the skin, but we see this also in the spleen. It could be that the host is redirecting defensive responses to be able to manage or cope with these two pathogens, or maybe the host is overwhelmed and cannot cope with the two pathogens. But it also is possible that this is pathogen mediated and that there's immunosuppression of different pathways in the salamander immune system or the immune response based on the presence of the fungus. And there's some evidence from lab studies that that's actually a possibility. Okay, the outcome, of course, of this immunosuppression is not good in terms of disease. These are the results from Anna's paper. And you can see that BD alone and control tend to have a high probability of survival for these salamanders that tolerate BD. If you infect them with B-sal alone, survival decreases significantly. So you have a 60% mortality, 40% probability of survival, and then the co-infected salamanders, that mortality just goes way up and the probability of survival is less than 20%. So not a good sign in terms of BD and BSAL. And I think there's some more that we can look at here, looking at sort of the order of infection and other things in future studies. Okay, I'm going to tell you one more quick story about differential gene expression. And this has to do with the two lineages of BD that exist in Brazil. So again, we have predictions. If BD-GPL is this highly virulent lineage that moved all over the world, we would expect higher disease severity outcome, right? And if BD Brazil has evolved in Brazil and has a long coevolutionary history with the Brazilian frog fauna, we might expect the evolution of some form of tolerance, right? Or some form of coevolutionary equilibrium. So our goal here was to actually look at how a single host would respond to both this new invasive hypervirulent form as well as the BD Brazil enzootic form. And the species that we used is this right here, pumpkin toadlet. It's the frog species with the cutest common name that I know of. Okay, again, we did an infection experiment on pumpkin toadlets, and this time we had control animals infected only with BD Brazil that survived at 100%, and then we had animals infected with BD GPL, and you can see that their survival was somewhat lower. In terms of how much they actually got infected and how much BD was in their skin, all the pumpkin toadlets infected with BD GPL had higher infection rates, higher loads than the pumpkin toadlets infected with BD Brazil. So BD Brazil does infect still, it doesn't kill, but it still resides in the skin, okay? All right, just quickly what we found, many more genes are differentially expressed in response to BD-GPL, this hypervirulent recently evolved lineage. And so you can see whether you measure gene expression early we did gene expression at 3 different stages of the infection process. Whether you do early, mid-infection, or late in the infection, the number of genes expressed by frogs infected with the 2 different lineages switches. Okay, so basically, frogs infected with BD Brazil, pumpkin toadlets infected with BD Brazil don't seem to express genes very quickly early on. By mid-infection, they're mounting their immune response. And by late infection, they've resolved that immune response, but the animals infected by BD-GPL are then going crazy. So they're increasing their gene expression relatively late in the game. And as we saw in the previous slide, to no effect, right? Because mortality is actually higher. So something interesting is going on here. Many more genes are differentially expressed in response to BD-GPL. And you can see that not only from these Venn diagrams, but also from looking at the overall gene expression rates in early, mid, and late infection. And you can see the characteristic, this is BD Brazil infected frogs. They're mounting their immune response right in the middle of the sampling period, and BD-GPL animals are only responding late. So here's another way to look at this. Let's see, okay, I'll walk you through this. So what we did was we took all those genes that were differentially expressed and binned them according to their functional categories. And then we can compare how frogs infected with BD Brazil and frogs infected with BD GPL are responding at each of the 3 sampling periods. Now, let me just back up a little bit and tell you that the reason that it's important to look at early, mid, and late is because the efficacy of an immune response, right, is dictated by basically the timing of different aspects of the response, right? So the innate immune system is the first line of defense. Certain things happen, certain pathways get activated, and those in turn have a role in terms of communicating, so the next step needs to happen, right? And so if there's any sort of dysfunction, right, in terms of what part of the immune response is getting activated at different points in time, you would predict that that could have fairly negative consequences in terms of the disease progression, right? Or the ability of the organism to fight the disease. So here's what happens with frogs who get infected with BD Brazil. Okay, early on, almost nothing happens, right? We don't see a lot of differentially expressed compared to the controls. Their genes getting expressed, they're just not largely or hugely differentially expressed. By the time you get to mid-infection, frogs start showing these 5 bars here, differential expression of key immune genes that are basically mounting this, this defense. And the immune genes have to do with this category here, collagen and keratin. So this is activation of keratin pathways, which are involved in skin formation, wound healing, responses to the skin in general. And so they differentially express genes in that pathway. They also differentially express a number of genes that are in various innate and adaptive immune responses. So exactly what you would like, right, to see an immune response that's ramped up. By the time late infection comes around, those frogs infected with BD Brazil have basically resolved the immune response and have no differentially expressed genes anymore. Look at what happens frogs exposed to BD GPL. Early on, they have some differential upregulation of genes having to do with immune responses. By the time mid-infection comes around, not much is going on. It's almost like the immunosuppression has already started. And by the time we get to late infection, a number of really important groups of genes are underexpressed, including this keratin pathway functional group. Okay, so again, downregulation of genes that are critical to immune response, just as we saw in the co-infected salamanders. Okay, so one thing that was interesting here is that I don't think we fully expected such a large difference between lineages of BD in terms of the infection outcome. But one thing that was interesting is that it looks pretty convincing that frogs, Brazilian frogs that evolved with BD Brazil actually have some form of successful immune response with the correct timing and the epithelial turnover, and basically they survive even though they're infected. This is what we call tolerance, right? So there's the ability of the immune system of the organism to somehow limit the damage of the pathogen that it has. Frogs infected with BD-GPL, right, a lineage that they did not evolve with, have this delayed immune response, late stage magnification, and basically dysregulation, which results in a form of immunosuppression. Okay, great. I'm going to stop here, but I just wanted to conclude by saying that a lot of what has driven sort of the questions that I'm interested in terms of organisms has to do with thinking about these traits. In this case, you know, either reproductive traits or immune traits or traits having to do with susceptibility to pathogens. I'm particularly interested in placing those traits in some sort of environmental context and trying to get at mechanisms of selection on those traits. And we do that often by sort of measuring outcomes, measuring function, either fitness in terms of my reproductive work and the work on studying diversity, or outcome of diseases in the case of my conservation work. All right, I need to thank everybody who is part of all these studies. I definitely did not do any of this work alone. And I want to thank especially my fungal genomics colleagues and my colleagues who work on sexual selection on frogs. And I am happy to answer any questions. Thank you very much.
Thank you so much for the really lovely seminar. Please ask questions in the Q&A box, which you guys will find at the bottom of your screen. We already have one from Brandon LeBombard. He asks, could the outliers or the larger deviation of the species with the larger testes, I think from the first study, correlate with cuckolding behavior in the other males?
Cuckolding behavior. The larger deviation. Yeah, so he means I think Brandon means the ones that are high, that have the larger testes, right? So the total, I guess the total variance, if you have an exposed mating system where everybody is together and there's competition, male-male competition for access to female eggs, I think you would expect that variance because, yeah, cuckolding behavior would be one of the mechanisms that might drive species with exposed amplexus to actually have larger testes because of that degree of sperm competition. What I don't know how to explain as well is what's going on down at the bottom of that variance, which is the guys with the relatively small testes that are still exposed. Now, I've looked at all the species that are in that large range of testis size. A few of them that are actually in the small testis group are actually animals that have very extreme forms of territoriality. And so they're somehow avoiding sperm competition by basically excluding other males, right? So these are animals with physical combat and stuff like that. So that's also a possibility.
Yeah, that totally makes sense. So Rick Kessley says, great talk. Where is B-cell in the world if it is not here? What do you think the future of frogs in North America will be with respect to BD in B-cell?
Good question. So B-cell is widespread in Asia, and it's we think that's where it emerged from. It is moved into Western Europe, and it is present there, and that's where it was actually identified as a pathogen because the Europeans started noticing declines in their salamandrid populations. So they have a number of European salamanders in the family Salamandridae, which are the newts and the salamanders. They tend to be a little bit more, they tend to be susceptible to B-cell. What is the future of frogs in North America? Well, interestingly enough, people always ask me, how do you talk about this stuff and still feel positive about conservation? And I think it's because we do have these very worrisome situations like emerging diseases like B-cell that may arrive and wreak havoc. But at the same time, I see a lot of cases where there is either the potential for recovery or there's some form of mitigating factor. So, it's true, we've lost a lot of frogs to extinction worldwide. And I think in different areas, maybe that impact has been larger, but I think that there's a good possibility that if we continue to study and be able to understand some of these dynamics, that there may be ways in which one could help. So for example, genetically assisted breeding, captive breeding might be an interesting thing to look at, and some people are looking into that. Genetic modification of the pathogen itself is a really interesting possibility that I know that some people are working on. So I think there is a lot to learn and maybe more to do.
Thank you. Vajrabhata asks, sorry if I got your name wrong, is there any reason why the frogs infected with BD alone had a higher rate of survival than the control frogs?
Yeah, that was it was only a few individuals that died. I think it was a husbandry issue. So the two weren't significantly different, the control and the BD alone. But wait, which one are you talking about? Which study? Can I see here?
Yeah, it's hard for them to clarify because they have to.
Oh, okay. It's not the frogs. It's the salamander study. In the salamander study and in the frog study as well, actually, with the BD Brazil and the control, I had a higher survivor with BD Brazil than I did in the control. And then in the salamander study, we had higher survival with BD alone than with control. Those two weren't significantly different in either of those cases. So, we did have some mortality in the lab that was potentially some other pathogen or independent of the infection, of the BD infection itself.
Liam asks, inferences based on the BD phylogeny depend on the tree being properly rooted. How was it rooted, and how confident are you that the root is in the right place?
Yeah, great question. There's also an issue there, Liam, that has come up in discussion with a bunch of collaborators that has to do with the fact that it's pretty difficult to do this because the fungal genomes are so bizarre in terms of huge chunks where we have certain things going on like loss of heterozygosity and so on. But there is probably some good possibility that our phylogenetic reconstruction is could probably be done better if we actually accounted for the potential of really deep introgression. So some people actually are proposing that BD-GPL actually arose because of a very, very old hybridization event, in which case we either don't know who those parents are, or we haven't been able to identify that yet. Our tree was rooted with, what was it rooted with? I can't remember since I didn't do the analyses. You know, I'm not going to tell you because I don't want to tell you something wrong. It was either rooted with B cell or another chytrid outgroup, but I can't remember.
Liam will probably be at the happy hour, which you're all welcome to come to the happy hour. I'm going to drop that in the chat in a second. That's going to follow this. But Doug Woodham asks, or says, great talk, Kelly, thank you. A couple of questions. I'd be really interested to know if there are differences in skin microbiota at that mid time point that would help explain tolerance to BD Brazil versus susceptibility to BD GPL. Any ideas there?
Well, we didn't measure it, Doug. I think we do need that, you know, and it's like, and I actually, Anna was the one who did the, oh, you're talking about this frog one. Okay, so yeah, we did not collect skin microbiota. We brought these guys into the lab, and as you know, right, once they're in the lab, it's very, everything changes pretty rapidly in terms of skin microbiome. So it would be interesting to look at that, but we don't have any data on that. That's partly why we need a collaborator.
So Doug's second question, is the Todd hypothesis about parasites being responsible for amphibian life history diversity fraught with the same problems as predators being the cause?
Yeah, good one. Yeah, possibly. Yeah, I mean, you know, it's like anything else. There's a possibility that attributing all variation to one cause is probably not a great idea, especially if we don't examine what other potential selective mechanisms might be. So
I think always both, all, some. For our final question is from Maria Navarrete. She says, thank you for the great talk. What do you think about the recent rediscoveries of certain species that were thought to have declined due to BD, such as Atelopus?
Yeah.
Are they resistant?
Yes, very exciting. There's two possibilities. Either they're resistant, in which case we should be able to measure that resistance with some transcriptomic work, or they happen to be in some sort of refugia that allowed them to survive. And in that case, the situation is a little more tenuous, right? Because refugia can be temporary as well. So those are the two possibilities.
One more question. Looks like it got in under the wire. And then we'll head over to the happy hour with, please feel free to take a break in between showing up there and now. But Molly Yu asks in her last question, did you look at male parental care in any of your sperm competition analyses? And if so, did it influence them?
We didn't find any association between parental care and terrestriality, but I didn't look at parental care and testis size. We don't have a full data set for the testis size. It's something that we're actively working on right now. So we're hoping to get data for all 200 species so that we can do sort of a more controlled phylogenetic analysis. And that's one of the questions that we'll be looking at, is because there's in Leptodactylus, there are cases where you have male parental care or female parental care. So both of those might actually be interesting.
Well, if you have any more questions for Kelly, please come to the happy hour if you can, and you can ask them there. Kelly, please take your time to come over there. We'll see you in a few minutes. The details for that are in the chat here right now. Okay. And we'll see you all there. Thank you so much.
Thank you.
Thank you. Thank you, Brooke. Great talk.
Oh, thank you.